Bone marrow microenvironment is an extremely sophisticated and highly coordinated niche for hematopoietic stem cells. There are multiple components of this. We have blood vessels, we have perivascular cells, stromal cells, fat cells, nerve cells, and so many more. And all these components govern and dictate the fate of an hematopoietic stem cell. Hematopoietic stem cells have the potential to create and regenerate all the blood cells in our body. My lab works on specialized bone marrow microenvironments, which govern these hematopoietic stem cells in different craniofacial bones in our head. So before I started my lab, I discovered that the skull bone marrow microenvironment is very different from other bone marrow compartments. But there are 23 different craniofacial bones in our head. So my lab is trying to find out how functionally unique and specialized these bone marrow microenvironments are. And we're trying to ultimately figure out why we have craniofacial bone marrow and why we need to have so many of them. As you know, we have severe bone loss during aging. But it's not only the bone that ages, the bone marrow also ages. We lose blood vessels in the bone marrow as we age. There's an excessive accumulation of fat cells, which directly inhibit normal hematopoietic stem cell activity, and the environment becomes very inflammatory during aging. Most of these studies have been done in long bones, in our arms and legs, and I found that the skull bone marrow is actually going in the opposite direction. It's actually growing and staying healthy and function. We're using innovative imaging methods like in vivo labeling with fluorescence conjugated antibodies to image these different craniofacial bones in its entirety. And we're using skull transplantation to assess specialized contribution from different craniofacial bones. We're also using intravital imaging to track tissue morphogenesis, and we're using also very creative methods of partial irradiation to assess their hematopoietic contribution to systemic circulation. Now we know that the skull bone marrow is continuously in communication with what's happening in the CNS, in our brain. We don't know yet which bone marrow microenvironments are contributing to the neuroinflammation and subsequent neurodegeneration. So we're hoping to find novel therapeutic targets within craniofacial bone marrow that we could employ to treat neurodegenerative diseases like Alzheimer's or neuroinflammatory diseases like multiple sclerosis.